everyone. It's my pleasure to introduce Steve Ragsdale, our speaker today. Steve and I share quite a few different locations. So Steve started out as an assistant professor in the Department of Chemistry at the University of Wisconsin-Milwaukee. And then in 1991, he moved to the Department of Biochemistry at the University of Nebraska. I graduated from the University of Nebraska in 1986, so I just missed him there. In 2007, he rose through the ranks there, became the Charles Bessey Professor at the University of Nebraska. And then in 2007, he moved to the University of Michigan. I got my PhD at the University of Michigan in '91. Steve has won many nice awards, and I want to point out that he's won several teaching awards, which is really fantastic because both at the University of Michigan as well as at the University of Nebraska. Steve, since the start of his research, has really been kind of at the forefront of carbon dioxide and carbon monoxide. And of course, now it's super important because of climate change. And so his research on the basics of biochemistry and how organisms fix carbon monoxide and carbon dioxide is really core to a lot of the things that we're doing here, as well as core to kind of the Biden administration's plan on fixing and reducing the amount of carbon dioxide in the atmosphere. So I hope you'll tell us all about how to do this and how we can solve this problem really quickly. Welcome to Jamie.
Thank you, Jay. Yeah, so thanks so much for inviting me to come here. I've known about this institute for quite a while, and I've been wanting to visit. So now it's really exciting to get to speak here and talk to you about some of our recent work. Okay, so what I want to do is talk to you about carbon dioxide fixation by anaerobic microbes. And so I'm really interested in the basic biochemistry of that. I've been working on it for many, many years, but I'm also interested in what we might be able to use this pathway for in terms of thinking about ways that we might deal with issues related to climate change. So I'll try to make that link as I go through my lecture. There's various things going on in my laboratory. The one at the top right is what I'll be talking about here. It's called the Wood-Youngdahl pathway, which is a major pathway for anaerobic carbon dioxide fixation. We work on methylation of mercury, but we're also interested in methyl coenzyme M reductase, which is the person who makes the other major greenhouse gas, methane. And it makes up maybe 70% of all the methane we have on Earth. And so those are the two major gases in the atmosphere. And then we do a lot of work also on regulation of metabolism in humans by heme carbon monoxide and redox. So there's the carbon monoxide link, which got me into working on human metabolism. So I know you've all seen these kind of pictures about CO2 levels and methane levels in the atmosphere. What I wanted to do with this, we can see them going back, thousands, tens of thousands, hundreds of thousands of years. But I just wanted to point out that we're still going up, and we're still going up exponentially, both in terms of CO2 and in methane. This last year was really a big amount. So you look down at the bottom, this is the growth rate of CO2. So the growth rate is also increasing, which is what you expect for an exponential function.
So may I ask what happened between 2000 and 2005 in methane?
What was happening at that point? I'm not sure.
Okay.
I'm not sure why it had that dip.
Yeah, it's interesting.
It is, yeah. Well, it had zero growth during that time.
Right.
It was still, we have a lot of methane in the atmosphere at that point. And why it didn't continue to grow. This has been interesting, though. You see almost a linear function for CO2 growth. Yeah. And methane has a kind of interesting kind of dip pattern, but it's then coming up again here. So those are things that are concerning. If you look at what's in the air, it's really kind of interesting. You hear about CO2 and methane, these greenhouse gases all the time. But actually, when you look at what's in the air, we have this little sliver down here that represents the greenhouse gases. And CO2 is 0.047% of the atmosphere, but it's too much. there's a lot of carbon, not only in the atmosphere, but in the earth in general. And so I think that that offers an opportunity for us because the carbon flux on our planet is huge relative to the small amount that's in the atmosphere. And so I feel like that offers an opportunity to use biology, which is responsible for all this global CO2 movement and methane movement, all the movement of carbon, to tweak, but potentially tweak things, whether it's nutritional or some deal with requirements and tweak the levels so that we move more toward the sinks, so that we can increase the level of sinks. So I'll talk more about that. It's really related to what I'm writing here. Okay, so we have the global emissions this last year again increased. Well, they were a record again, like it is every year. And this last climate change conference, this was an interesting statement for two reasons. So I'll just read, you can just see what's up here. Human-induced climate change is causing disruptions affecting the lives of billions of people around the world, mainly people who are least equipped to handle it. And to avoid this mounting loss of life, biodiversity, and infrastructure, urgent action is required to adapt. So now they're talking about adaptation. It's now really clear what is ahead of us. So we have to find ways sociologically, religiously, in all our ways of life. The IPCC is saying we need to learn to adapt to that, as well as make deep cuts in greenhouse gas emissions. Okay, I agree with those things. On the other hand, the lifetime of CO2 already in the atmosphere, 380 billion tons, is 300 to 1,000 years. So if we stop right now with our emissions, that CO2 is still in the atmosphere for all that time. And methane is about 10 years. 2 billion tons of methane, but in fact, that's 80 times more potent as a greenhouse gas. So that's like having another 160 billion tons out of the atmosphere. So besides emission cuts, it's really crucial that we develop rapid and efficient strategies to remove CO2 and methane from our atmosphere. That's what I think. That we have to have biological ways to do it, though. And they talk about, for many years, they've been talking about major CO2 mitigation strategies: carbon capture and utilization, and carbon capture and storage. Chemical ways, engineering ways to do that. But now, for the first time, the IPCC is also talking about nature. I mean, they're mainly talking about increasing the number of trees and not cutting down trees. But I think that there's a lot that we can do as microbiologists, as chemists, as engineers to work together. And I think that we can actually handle this. So if you just look at the overall sinks and sources of CO2, this is very simple. We have in red all those sinks of CO2, photosynthesis, plants taking up CO2, microbial metabolism taking up CO2. We have all these different ways for our sources of CO2. And this is, of course, this balance of sinks and sources. And right now, this fossil fuel is the major contribution that's taking us out of whack. And so we need to do something about that. So anyway, there's 6 natural biological CO2 fixation pathways. And what I wanted to emphasize here, these are all potential ways to deal with climate change, because there are all ways that organisms are taking CO2 out of the atmosphere. The Calvin cycle is the one we know the most about. Calvin, from right here in Berkeley, is the person who made the major efforts in discovering this pathway a long time ago, reductive citric acid cycle. And you can see there's a lot of ATP that's required in each of these pathways. So it's basically a very energy requiring pathway to take up CO2. And at the bottom, I'm showing you the Wood-Youngdahl pathway. And this is different in one of many respects. One of them is that it is the least energy requiring pathway. If you put them all on equal scales, you can see that the amount of ATP per 3 carbon sugar is less than in all these other pathways. And that's because these microbes are living right at the edge of life. They're not taking up they don't have ways to make lots and lots of CO2, excess CO2, and then expend that on CO2 fixation. They have to actually make a living fixing CO2, which they've learned how to do, which is remarkable. So in this, there's no extra ATP requirement. The enzymes are highly oxygen sensitive. Just pointing out some special characteristics of this that biochemists and microbiologists need to think about for the pathway. It's weird in that it is the only organometallic pathway we know about. Series of metal carbon bonds are formed in the pathway. It generates carbon monoxide as a metabolic intermediate, which sounds crazy since carbon monoxide is a toxic gas, but they have ways to deal with it internally so it never actually leaves the organism. And it would be very expensive if they lost it because it's an important source of carbon for them, for these microbes. It's a reversible pathway. Lots of organisms actually grow on acetyl-CoA. Others make acetyl-CoA. And it's thought to be the most ancient CO2 fixation pathway. Clearly, it was present in LUCA, last universal common ancestor, like nitrogenase. And organisms that use this pathway are able to grow on a lot of different compounds. So this shows, it's amazing. They take a carbon monoxide, hydrogen, CO2, they take all these compounds up, metabolize them. They have a lot of electron acceptors besides CO2 that they can utilize, and they can make a lot of products. And so that allows them to have a metabolic machinery that if you can figure out good ways to optimize CO2 uptake, then you already have a pre-existing pathways there to make a lot of compounds that are very interesting from the standpoint of biotech. But they also can be engineered so that you can put in new pathways and use those reducing equivalents and use acetyl-CoA, which is at the very center of metabolism. Use that acetyl-CoA to go in all kinds of directions. So it's remarkable that what I feel is the possibilities for this pathway. So this is just an overview of this. And there are really 4 cycles that are coordinated. So there are 2 CO2s, and I've colored them here, red and blue. And they end up forming the 2 carbons of acetyl-CoA. So it's a very unusual pathway of acetyl-CoA formation. And in this, I'm showing you the metal centers in these enzymes, which are really key to the pathway. I've mentioned that it's an organometallic pathway, and those intermediates in the pathway are strictly enzyme bound. They're not soluble, but they're enzyme bound, and they're bound to metal centers in proteins, which is why it has taken so long to characterize this pathway. Whereas the Calvin cycle was characterized in the 70s and earlier. This pathway, we've only just this year finished isolating the key intermediates in the pathway. I told NIH I was going to do this though in 1987, in 5 years. I didn't realize what was ahead. So to go through the pathway kind of quickly, we have first carbon dioxide uptake pathway, first carbon dioxide uptake cycle. It can be linked to a lot of electron donors, like hydrogen or other donors, and is converted to carbon monoxide. The other carbon dioxide is converted to formate, and that enters another cycle of folate-dependent enzymes to make a methyl group. That methyl group then is transferred to B12 and forms the first of that methyl branch of organometallic intermediates. And then an enzyme, acetyl-CoA synthase, takes the carbon monoxide onto another center, and it moves it around this pathway. I'm pointing with my finger at myself here. And then the methyl group hops on this, what we call the A cluster, a nickel-containing iron sulfur cluster within the protein, makes an acetyl intermediate that just reacts with the CoA and makes acetyl-CoA. So it's really looks pretty simple from an inorganic standpoint. The pathway is exergonic, so they can make 95 kilojoules per mole, so they can actually then use electron transport related phosphorylation to generate ATP for their cells. And this is the folate pathway. I thought I was going to show that earlier, but this is how CO2 gets to methyl tetrahydrofolate over here. Oh, I have to do it like this. So a lot of folate enzymology, but all of those are enzyme-bound intermediates as well. So you'll never find that an organic intermediate in solution for any of these intermediates unless you unless you got the protein in hand and have ways to characterize those intermediates directly on the enzyme. So here's the structure of the key complex. And I was showing you carbon monoxide dehydrogenase and acetyl-CoA synthase. So this forms this butterfly-shaped molecule in the middle where carbon dioxide is reduced to CO. It has 5 metal clusters in the middle. And the one that's most important from the standpoint of catalysis in terms of CO2 reduction is this cluster called the C cluster that's in green. And that binds carbon monoxide and undergoes reduction. And then it then travels to this protein that's attached to it. And that is where the carbon monoxide is further metabolized to make acetyl-CoA. There's also an electron transfer chain in this protein, carbon monoxide dehydrogenase. To make electron transfer fast, it's an extremely rapid reaction. And from the standpoint of CO2 reduction, it is at the diffusion limited rate. So extremely fast chemistry through electron transfer, as well as the carbon transformation to make carbon monoxide. And we'd like to understand more about how that happens, because chemists are working like crazy to figure out how to reduce CO2. And not only is CO2 thermodynamically difficult to reduce, most catalysts actually would rather reduce protons because it's such an uphill reaction. So generally the catalysts, they're always fighting the production of hydrogen when they're trying to reduce CO2. This enzyme won't reduce protons to hydrogen. Not only that, the thing the chemists are really struggling with is that in CO2 reduction, it always goes through, not always, but predominantly, the way that these catalysts work is they do one electron chemistry, and they have to go through a radical anion, CO2 radical anion. And that is way, way uphill. And so that is a huge barrier. You have to put in an extra volt to be able to do that reaction. Whereas the enzyme actually knows how to do the reaction at its thermodynamic potential. It's low, minus 550 millivolts, but it is in equilibrium on the enzyme. So somehow the enzyme has figured out how to run this reaction without going through that radical anion, and to do it at its thermodynamic potential, and to do it fast, and at the environmental concentrations, at ambient concentrations of CO2. That's the other major hurdle for any carbon capture. Some people say it's not even feasible because you've got to concentrate the CO2 so much. Well, here the organisms are taking it right out of the air and converting it right into a major chemical feedstock, acetyl-CoA. So I mentioned CO, carbon monoxide, is a toxic gas, but it turns out that these two enzymes are so beautifully connected that they have a tunnel that runs between the two active sites. So when carbon monoxide is made, so what we did here in collaboration with Kathy Drennan, we crystallized the protein, but then we put it under xenon gas, really high pressures of xenon gas. And xenon's a heavy metal. This is often used to find channels in proteins. And so we did that with this protein. And what you see here in blue are the xenon molecules that were trapped. And so with that, we did some computational work, both on the left and right more recently. And what we can see is that this channel runs between the site of carbon monoxide formation from CO2 in either direction down to acetyl-CoA synthase where it's utilized. Over here, same thing. And it turns out if you take a labeled CO2 and you put it in carbon monoxide without a label and look for the acetyl-CoA that's formed, you'd expect that carbon monoxide, this little thing, would be in equilibrium with a solution. But even at 1 millimolar CO, as high a concentration as you can get in solution, there's absolutely no exchange. This channel is so tight that it migrates through the protein without any loss of carbon. And that's really essential because this is such an uphill reaction for the organism. If it lost that, then it couldn't live. And I'll tell you some experiments we've done that test that idea. And there's several ways that they maintain that carbon monoxide. That one of them is extremely surprising. We've done some recent modeling with a group from PNNL, Simone Roger, and this is all molecular dynamic simulation. So we can recapitulate what we saw on the crystal structure, but also we can see that there is a lot of motion within the protein, but you can also see that the carbon monoxide from the molecular dynamic standpoint never gets close to the solution. So we are spending time trying to understand more about that. So I wanted to just kind of run through some recent studies, because in 1987, we hypothesized that this pathway was organometallic based on the fact that we had a nickel enzyme and we could see an EPR signal from nickel. But then we found out that the protein also has iron. And anyway, at that point, we hypothesized that this was organometallic nickel chemistry, because all we knew the protein had at the time was nickel. And then for the last 30 some odd years, I've been actually wanting to validate or disprove that hypothesis. And it's just in the last 3 years, we've been able to capture those intermediates using probably the only method that would have allowed us to see this. I've tried so many methods to visualize this, but it's not sensitive to Raman. And most of the techniques you would want to use to look at metal carbon bonds. What we ended up doing was using X-ray absorption spectroscopy with Ariti Mukta Sarangi, who's here at SLAC. It's a wonderful X-ray absorption person. If you ever wanted to do that kind of experiment, to look at a metal center with high resolution, then she's amazing. And then with Kathy Drennan, crystallography of one of the intermediates we were able to accomplish recently. So I think I'm going to just kind of go through this. There's just a lot of things that we did to show how complicated the cluster is. And that it's not just a nickel-based center, but it's a highly delocalized cluster. Electrons are spinning all over the place in the cluster. It was key to us that whatever we capture, we find some way to visualize an intermediate that we know how fast it happens, how fast it forms, and how fast it decays. And that's called a test of catalytic competence. And so we've done that for all these intermediates to validate that. The first intermediate that we were able to trap was the carbonylated intermediate. So at the end of the channel, then there's this metal cluster called the A cluster. And we used a technique called Zanes and another technique called EXAFs. And this is a technique that puts high energy X-rays into the sample of either crystalline protein or solution of protein, and it excites electrons. They come out of the protein and they bounce around actually and hit the ligands that are surrounding the metal center. So you can go right to the nickel center, you can shoot electrons at it, it will hit those atoms on the outside, and then you can back calculate what those ligands are and what their distances are. And that allows us then for the first time to see the carbonylated intermediate. So this is what we did in 2017, and it gives you really beautiful resolution on the distances. And for the first time, we could see the metal carbonyl. And it's a nickel carbonyl. And then a couple of years later, in collaboration with Kathy, we were able to crystallize that complex. So we have good high resolution pictures of how carbon monoxide binds. Then we were doing some other experiments. And what we were doing is flashing off carbon monoxide by hitting it with a laser and hitting right in the 400 nanometer region where the absorption for the metal carbonyl would be. Not the metal carbonyl, but the metal center itself, the iron and nickel sulfur bonds in the center where it's so delocalized. At any rate, we were using high energy laser to photolize off the carbon monoxide. And we found an intermediate, which is, a really important intermediate that we've never been able to trap otherwise. But the interesting thing is that we could only do this at less than 50 degrees Kelvin. If we photolize it higher than that, it comes back on so fast we never see it leave. And so we did this with a lot of different experiments, looking to see the photolysis and the rebinding. You could see infrared spectra from CO. We could see the EPR spectra at different temperatures. And what we found was that the free energy for recombination of that CO is even tighter than that for hemoglobin. It's like 1 kilojoule per mole. And it was shocking because that is almost no energy. It's like it's on the protein and it's off the protein. The picosecond you flash it off, it's going to rebind. So we got to go to really low temperatures to be able to see the dissociated form of the protein. But what that also meant was this: what is holding it there? What on earth is keeping it so close? And we noticed on the crystal structure around that CO was something we called an alcove. There were a bunch of residues around the xenon. And if we looked at that, they were aromatics. And in fact, if we went through and looked at each one of these little xenons, every one of them are surrounded by little boxes. And it's almost like you've got a ping pong machine, what am I trying to? Pinball machine. I guess it could be ping pong machine too, but that's not what I was looking for. Okay, anyway, so and this pinball machine is just taking these CO molecules and shooting them down into each one of these boxes, and it's moving on. There's a lot of dynamics, as we've seen with the molecular dynamics simulations. But we presumed that this was, and hypothesized that this was what was holding the CO so close to the metal center that it would bounce off and bounce back on. There's some important mechanistic reasons that this protein does that. What I wanted to do, I was thinking that, well, okay, what if we kill this site? What if we take one of those walls that's surrounding the xenon right by the nickel center where the catalysis takes place? What if we convert one of these phenylalanine residues, which we chose phenylalanine 212, convert it to alanine? Then we would open up a wall and we would presume that then the CO could escape for the first time. And so we did some experiments and could see that with the alanine mutation, we could see that, well, first I'll just look at the wild type. So the wild type protein has a pretty decent binding constant of 12 micromolar. So the way we do this is EPR spectroscopy. We put the protein with CO, freeze it, and then run an electron paramagnetic resonance spectrum. And that is paramagnetic, so we can see a CO-dependent signal formation. So each one of these down here, this is no CO, then a little bit of CO, we keep increasing. And from that, we can get a KD. So 12 micromolar, which is it's pretty reasonable for organisms growing under CO2 and generating a local amount of CO. But then when we turn it into alanine, then we see this huge difference of about 20-fold. So that now the KD for CO, we can't even get there. We can't even get to a complete saturation because the site is now opened up to the point that it doesn't stick to that protein anymore. And then we wanted to see, well, so what does that do to the organism? And in a collaboration with Lanzatech, what we found was that when we look at the organism growing on fructose, growing on heterotrophic conditions, they can still grow just fine. But as soon as we put them on hydrogen CO2 and we take this mutant that has the alanine, but on this lane right here, I'll just do it on the side, then that F2209A will not grow autotrophically at all. So what that means is that alcove is absolutely essential for autotrophic growth for these organisms. It's not just the metal center, it's actually this amazing cluster. alcove or a cage that sits right by the active site and compresses CO near the active site and prevents it from escaping from the protein. So we were thinking, okay, we know how to kill it, but what if we try to make it better? Because the organisms have grown under conditions that are required for it to grow. They can make enough CO to do that. But in fact, to optimize their growth, what Lanzatech does is to grow with a little bit of carbon monoxide. Carbon monoxide is not cheap and it's also dangerous. So what if we could just lower the levels of CO? So we have a phenylalanine we were playing around with. What happens if we turn it into a tryptophan and make a larger residue? And those are the only two we tried. We tried both extremes. And what we find when we now do the CO titration is that now it goes to picomolar binding constant for CO. So I find that remarkable. But I thought, well, maybe that would kill its activity for the other reactions. Because the protein has to not only carbonylate, but it has to go through a methylation cycle. CoA has to bind. And I know that CO and CoA are competitive, but in fact, there's no effect on the overall rate. So all it seems to have happened is the Kcat over Km for CO has gone up astronomically relative to the wild type protein. So there's a lot we want to do to examine the implications of that. But the protein has this interesting characteristic that it can either bind carbon monoxide or it can bind a methyl group. It has this random aspect to its mechanism, which is great for a biochemist because now we can also look for that methylated intermediate using the same techniques. And so again, in collaboration with Riti. So what this is going to show you is the choreography around the protein. Because this protein has to bind a methyl group, but it's also got to bind another protein. It's also got to bind CO. And so the acetyl-CoA synthase that I was just showing you, the active center is shown here at the top. The methyl group is coming from methyl tetrahydrofolate. And then it's converted, and then it's going to bind to this coronoid iron sulfur protein, which is a B12 center. And then that is what gets transferred to this protein. So there's a lot of conformational movement that has to take place. That's just represented here by some kind of sticks and stones and stuff, rather than the actual crystal structure. We have structures of those complexes. Okay, so first you can see the methyltransferase binding, methyl tetrahydrofolate binding to B12. It protects it in a little pocket, and then it transfers it to acetyl-CoA synthase. It's just a cute little picture to see. But there really is tremendous movement that has to take place within the protein. So these little pictures are actually substantiated by detailed X-ray crystallographic studies as well. Okay, so the nickel methyl intermediate we've been able to characterize, again, by these methods, could substantiate the loss of the methyl group from the cobalt as it's transferred to the nickel. And we can see these big changes in the nickel spectroscopy relative to the iron, which has, almost no changes when you do that. And from that, then we can get all the details of the coordination geometry for the methyl group. There's been a few other postulates that maybe methyl binds to a sulfur, maybe methyl binds to an iron, but we can clearly see this nickel methyl group. And interestingly, it binds within the plane of the nickel ion, the coordination sphere, whereas the CO binds tetrahedrally to protein. And we've also isolated the acetylated intermediate, substantiating that part of the pathway. And there's some changes, but it also stays square planar. So we've trapped these intermediates, and I'm not going to go into any more detail other than just say that now we've got this pathway showing this organometallic pathway in hundreds of microbiology books and chemistry books. And they're the basis of a lot of people's research grants, that this is the way the path works. And for the last 30 years, I've been wondering, it would have been exciting maybe if it wasn't what we had originally proposed, but it just feels nice finally to understand that this really is a pathway that involves organometallic chemistry. So the pathway involves first a reductive activation. We cannot reduce the protein to this, we can't reduce the protein to this reduced state without coupling it to substrate binding. And this is a really interesting thing that we found in this whole pathway. The major catalytic intermediates seem to be shielded so that they go into its active state, and that's in equilibrium with a protected state. And as it binds the next substrate, the active intermediate gets re-recruited back into the catalytic cycle. So it's called a chemical coupling mechanism, and it's not that uncommon for electrochemists or in chemical catalysis to see this happen. But for every intermediate, we can't reduce the protein without CO, but then we reduce it quite easily. If we put in the methyl group, again, we can't reduce it and get it to bind methyl. You have to have a reductive activation, but then the binding of the methyl group pulls it forward into another very unstable intermediate that's highly oxidized. And then that's in equilibrium with another intermediate that's stable enough to maintain without it reacting further with some other stuff in the active site, like hydroxide ions. And then carbon monoxide can bind to that site, making the acetyl intermediate. And then from there, coenzyme A will bind and excise that carbonyl group to make acetyl-CoA. So it's a quite simple pathway in a way. I mean, just thinking about what it does, a single nickel center seems to be at the heart of all the catalysis. And all it's doing is binding little one carbon compounds. So we should have been able to isolate it years ago. Okay, but anyway, and in summary, this pathway involves this novel enzymatic system in which we have enzyme-bound and nickel-bound organometallic intermediates. Know that the pathway requires an alcove for CO binding and also for autotrophic growth. This is a pathway that utilizes organometallic enzymology, gas channels, a CO alcove, protein-protein complexes, large conformational changes, and this electrochemical chemical coupling that allows this pathway to exist and to finally coordinate all these changes that are taking place only when substrates are present, to allow it to react with the substrates when they're in line, and to then shield those intermediates before the substrate shows up. So there's a lot of things we still want to do. We still don't understand how coenzyme A binds to the protein. We have some evidence that it also binds to nickel. We're doing a lot of computational chemistry to model, especially these transient species that I don't think we'll ever actually characterize in solution because they're so reactive that even using millisecond type mixing methods, we've never seen them. So we'll need to rely a lot on the computational chemists, and then we can make predictions based on what they find, that we can then go and test those and work really closely with our group at PNNL to verify the computations. There's another complex we've not yet crystallized. Kathy and I are working hard on that. And since I've been here at Berkeley, it's the main reason I came here with one of my students, is we're using serial crystallography, which is an absolutely amazing technique that Jan Kern, Junko Yano, and Vital Yachandra up on the Hill have developed, along with some others, but they've made really huge advances in this. I've been watching it for the last 15 years, hoping that it gets to the point that we can start studying oxygen-sensitive proteins. And that happened 2 years ago when I got a call from Vital saying, okay, I want to look at your proteins. And so we started a collaboration both on this protein and on methyl coenzyme M reductase. But if you get a chance to hear them talk about that technique, it's absolutely stunning. What it does, in effect, I've been using transient kinetics for many years, where you mix two things together in a stop flow, and you get a profile, a spectrum from that. Well, now with XFEL, you get a structure. It's a rapid mix structure of intermediates. They're femtosecond pulses, and those femtosecond pulses destroy your crystal, but it's a method that's considered to be a detect before destruction method. Like you can see the crystals just get vaporized. But because it's only a 10 to 30 femtosecond pulse, it's not enough time for the radicals that happen within the sample to go around and start destroying things like metal centers. And so personally, I think that the structure of every redox active metal center is probably not completely right, because crystallographers put these proteins, we do it too, we put these proteins in high intensity X-rays and keep collecting data for maybe 30 minutes. And in that time, there are all kinds of things that happen. And studying one of our proteins that has a really beautiful chemical change that you can assay to see these changes, we can see them happen within 10 shots with an X-ray. It's already destroyed the metal center, even though the protein absolutely looks fine. Overall, you wouldn't know that it's pockmarked with hydroxyl radicals and the metal center has been destroyed. So anyway, I'm super excited about it. And I think it's going to be a really important development for biochemistry and for understanding mechanisms to get the true structures of these proteins. And I do feel that this is one pathway, and there are others that you guys are working on that can have huge effects on greenhouse gases and removal of those greenhouse gases from our environment. And we can think about potentially bioengineering the planet in a way. And it's a scary thing to use those terms, but what have we done for all these years other than bioengineering the planet to put us out of balance? So at any rate, I feel like these sort of pathways, developing a better understanding of exactly how they work is really key to our future and creating carbon neutral or carbon negative type environment right now to help reach the right sort of balance on Earth. So thank you so much for your attention. These are the people who've been working in my laboratory. We've been funded by NIH and Department of Energy, and a grant with RPE and Lanzatech to do some of our work. So it's super exciting. They're wonderful people, a lot of exciting things going on right now.
I noticed an R35 on your NIH. So even though you proposed that you're going to find the mechanism in 5 years, they just kept funding you?
They just kept funding me all these years. We kept coming up with things that they found exciting, even though like that bottom line that was in the back of my mind was still out there.
For those of you who don't know what that means 35 years on basically continuations, right?
Yeah, it has been going on for 35 years.
Competitive, right? But still,
Yeah,
that's impressive. You engineered one of the boxes or whatever with an additional aromatic, and you got the KD to be, if I'm right, extremely low, right? You dropped it even. Why didn't that evolve that way?
I don't think it needed to. I think that if it was so effective at making CO from CO2 that actually didn't need to go beyond this like 12 micromolar. That's my personal opinion. I feel like with most of these systems, they're not really optimized, and that we can re-optimize them by giving them new selective pressures and optimize them toward a particular biotechnological goal, for example, that may be much more rigid than the organism would need for survival.
But having that very low KD doesn't hurt the process in some way, like the CO won't move out of it?
I would have thought so. But the CO doesn't move out until it makes acetyl-CoA.
Got it.
That's the key. You don't want it to get out. Once you make it, you want it to get through that channel, a tight channel. You want it to get to the active site. And if it ever comes off the metal center, which it might even have to do if it's going to bind a methyl group, but then it'll pop back on again because there's no free energy. It's just like it's actually an extended active site.
You do have an online question from young Kim. A simple question: are those enzymes and proteins involved in the WLP membrane-bound or freely floating enzyme systems in the cytoplasm?
Well, I have to answer in between because they are not, none of them are membrane associated, which is great. Because that for the biochemist, it's great. But they do form complexes. And so CO dehydrogenase, acetyl-CoA synthase, those are two enzymes, but they form this tight complex. You even isolate them together. Methanogens, the whole 5-protein complex is isolated together, some of the methanogens. So I think in the cell, they actually all come together as a complex. But they do have to dissociate, as I was showing you in that little cartoon, that they do have to dissociate from each other and then come back on. So it's sort of in between. They are not free floating because they associate with other proteins, but they are not membrane bound. And you have a question.
Yeah, so could the acetyl-CoA synthetase be used in isolation? Let's say you feed the cell carbon monoxide rather than carbon dioxide. Could it function alone?
Yeah, there's actually 2 forms of carbon monoxide dehydrogenase. And one of them is associated with the hydrogenase pathway. And it turns carbon monoxide at the small levels that are found out in the atmosphere. It turns the carbon monoxide into CO2 as its physiological function. And the 2 electrons are linked that turns on as soon as CO is generated. As soon as CO is present, it turns on the pathway for the hydrogenases in a lot of organisms like Rhodospirillum. And in that case, it doesn't even bind to acetyl-CoA synthase, even though the overall structure of that complex looks almost identical. RMST maybe less than an angstrom for all the atoms in the protein. I mean, there are some substitutions, but overall that structure is highly maintained as that butterfly structure I showed you early on, but it performs the opposite function, CO2 oxidation instead of CO2 reduction, and is coupled to turning protons into H2 for the hydrogen pathway.
Are there like versions of these enzymes that are oxygen tolerant, or are both ACS and carbon monoxide dehydrogenase?
And not for this particular enzyme. There's some that are less oxygen sensitive than others. So Kathy Drennan, for example, is working with one from disulfovibrio that is less oxygen sensitive than the ones that I've been working with for so many years. And it can be reactivated. And that's something else I think is super interesting, because I actually think that there are ways that we can manufacture a protein or bioengineer a protein by playing around with the channels and also some of the metal clusters to make them oxygen insensitive. But that's, these organisms are actually ancient organisms, and they've made their whole life, like evolutionary life, in anaerobic conditions. And so this is another situation where they don't care, they just stay out of the oxygen. But for us, it's really a pain actually to work with anaerobic enzymes and to think about some of the biotechnological problems of maintaining strictly oxygen-free conditions when you're trying to run these pathways. Then it would be a whole lot easier if you didn't have to worry about a little bit of oxygen coming into the system and killing your whole fermentation process. So we're working some on that. There's some other people, around the world who are also working on making these oxygen insensitive. There's another protein that's a carbon monoxide dehydrogenase, though, that's a molybdopterin protein. And that one is a lot slower than these proteins, but it also is oxygen insensitive. So the molybdopterin protein doesn't even need reactivation. It works merely in the oxygen.
What's the mechanism of oxygen inactivation?
I think there's several things. One of them, the metal centers of all these proteins contain iron sulfur clusters. And those iron sulfur clusters are almost everywhere you see them. You'll see that they are highly oxygen sensitive, and they just start to fall apart, turn into rust. And so then you have to rebuild a metal cluster or rebuild a protein in order to start over again. That's one of the things. But there's some of the metal clusters are less sensitive than others. So there's one sitting right at the entrance where the electrons first come into the protein. And at least Kathy's shown that modulating the properties of that site, she can make it less oxygen sensitive to give it some protection. In some cases, it disulfides around the active site, that helps transfer some electrons and protect it. And then you can do a reduction. But yeah, that's probably the main mechanism is probably through iron sulfur cluster chemistry.
Well, thank you so much for a very exciting talk.
Well, thank you.